Hello students, now we will discuss the deficiency manifestations of vitamin B12 and folic acid. Very important vitamins. How will you differentiate between these two? Because the deficiency causes anemia, that too macrocytic, means megaloblastic anemia will be there in the deficiency of B12 and folic acid both. Okay, so macrocytosis is there, means increase in mean corpuscular volume. There is increase in mean corpuscular volume. Anemia means decrease in the hemoglobin level. And hypersegmented neutrophils, these are the features of megaloblastic anemia. So anemia, macrocytosis and hypersegmented neutrophils, because this B12 and folic acid, they are required, principally the folic acid, they are required for the synthesis of DNA and RNA. Nucleotide synthesis they required. So the cell growth is means cell division is arrested in the S phase of cell division, and cell grows continuously without cell division. So macrocytosis will be there. So for that, for differentiating the megaloblastic anemia, do the vitamin B12 level. If it is decreased, means if B12 level is low, means it is B12 deficiency. Do the folic acid level. If folic acid is low, means folic acid deficiency. Do the methylmalonic acid level. If methylmalonic acid levels are more and B12 levels are low, that indicates the B12 deficiency. Increase in methylmalonic acid level, that indicates the B12 deficiency. Okay, and there is CNS involvement, degeneration of spinal cord. All of you know that B12 is required for the conversion of methylmalonyl coenzyme A. For this reaction, methylmalonyl coenzyme A is converted into succinyl coenzyme A, and this reaction required B12. B12 is required. Methylmalonyl CoA mutase enzyme is there. Mutase enzyme is there. If there is deficiency of B12, there is accumulation of methylmalonic acid, and methylmalonic acid excreted in the urine. This methylmalonyl coenzyme A, it causes the defect in the fatty acid synthesis, is also causes the defective lipid synthesis, that is glycolipids, sphingolipid synthesis. It causes the defective lipid synthesis and defect in the myelin synthesis, myelin formation. So that causes the subacute combined degeneration of spinal cord. That's why CNS manifestations will be there in the vitamin B12 deficiency. Now folate. Folate, if there is folate deficiency, there is excretion of FIGLU in urine, that is farm immunoglutamic acid in the urine. This is done by the histidine load test. Histidine is given. Histidine is converted into FIGLU, that is farm-immunoglutamic acid, and it is converted into glutamate, L-glutamate. Okay, it is converted into glutamate. For this conversion, tetrahydrofolate is required. Folic acid is required. If there is folate deficiency, this FIGLU level will increase and it is excreted in the urine. FIGLU will excrete in the urine. So this is vitamin B9 deficiency or folate deficiency. And if the folate deficiency occurs in the pregnancy, it causes neural tube defect in the form of spina bifida and anencephaly. That's why the folate is given when the pregnant lady visit to the hospital. Folate is given. Folate and iron supplementation is given. Also, in folate deficiency, there is increased homocysteine level. So, homocystinemia is there. Increased homocysteine level causes aggregation of the LDL particle, and it is responsible for the cardiovascular manifestation in the form of thrombosis. So, these are the features of folate deficiency. Remember the FIGLU excretion in the urine. These are the features of B12 deficiency. Remember the methylmalonic acid excretion in the urine. So these are the sites for the absorption of iron, folic acid, B12, duodenum, proximal duodenum, jejunum and ileum. These are the sites for iron, folic acid and B12 absorption from the intestine. So keep watching. Thank you.